Human physiology is concerned with it is the study of specific characteristics and mechanisms of the human body, and that makes it a living being. So we have to study about the cell and its organelles, and how the body is functioning inside the system, that is how the nerves are functioning, and what's the basic mechanism of that, and what are the ionic changes happening during each action. So the organization of the human body consists of cells, tissues, organs, and organ system, and man in the environment. So the environment is divided into external environment and internal environment. So the external environment is the place where the man lives in. That is, there is so many factors are involved in the living process. So that is, there will be oxygen, water, food, physical factors, and social factors will be there. Even microorganisms and parasites will be present in the external environment. But the internal environment is things to be constant, means it is in a balanced position. So Claude Bernard, the scientist, states that all the life processes have only one goal, that is to keep the internal environment constant. So to keep the internal environment constant, that is known as physiology actually. All the body functions are happening to make the internal environment constant. And that process is known as homeostasis. Homeostasis is maintenance of static or constant conditions in the internal environment. Homeostasis. So there are two types of homeostatic control system. They are negative feedback mechanism and positive feedback mechanism. Negative feedback mechanism and positive feedback mechanism which act on our body to maintain the internal environment constant. So for each system, we have a receptor which detects the change in the body, a control center which determines a set point for the normal range. And there is an effector will be there, any organ which causes the response determined by the control center. This is the basic components of our feedback system. So if the activity of a particular system is increased or decreased, the control system initiates a negative feedback which returns the activity towards the normal. For example, negative feedback mechanisms, there are so many examples. Most of our body functions are under negative feedback mechanism, especially hormonal control. Almost all the endocrine system are under negative feedback mechanism. For example, here we are thinking about the insulin or the blood glucose level. So if the blood glucose level going high above a particular level, or if it is around 90 to 110, if it is going above 110 or 120, the insulin secretion will be more. And if it is coming down, there are so many other hormones like glucagon, growth hormone. So all these hormones will act, and it will make the system to the normal. And coming to the positive feedback mechanism is the mechanism by which the system will act increasingly. Means if there is a change or if there is a decrease, it will increase, the system will increases the action up to a physiological limit. So in case there are mainly three examples we can see, that is blood coagulation. So if there is a tear of the blood vessel, the clotting factors will reach there, and it will activate a cascade of reaction will happen. More and more and more of proteins and chemicals will be released, and it will seal the wound. till that sealing process happens, the action will be increased. Okay, that increased mechanism is the positive feedback mechanism. Other two examples, you have to remember, the other two examples are in the endocrine system. They are known as neuroendocrine reflexes. Neuroendocrine reflexes, they are parturition reflex. First one is parturition reflex and second one is milk ejection reflex. These are all the positive feedback reflexes and neuroendocrine reflexes in the body. So coming to the cell, that is the basic unit of any living organism, which has cell membrane, cytoplasm, and a nucleus. So cell membrane is the boundary of the cell, which is made up of phospholipid bilayer. So in this cell membrane, you can see it is around around 7 nanometer in thickness, that is the phospholipid bilayer, and which is having an integral protein and peripheral protein. We have to note this points, like integral protein will extend from one side to another, and the peripheral protein will place only the outside of the cell membranes or the cell membrane. Okay, so these are all the two protein molecules present in the cell membrane, integral protein and the peripheral protein. And there are carbohydrate moieties, cholesterol and protein, all those things will be present. You can see it in detail. And the proteins which present in the cell membrane are lipoprotein. They are the enzymes and ion channels, and glycoproteins, which are the receptors for hormones and neurotransmitters. They are lipoproteins and glycoprotein. Glycoproteins is which contains a carbohydrate moiety in that, so it is known as glycoprotein. And there are another protein types known as intrinsic proteins. They are mainly enzymes, and peripheral protein. They are the cytoskeleton. Cytoskeleton means which gives a structural integrity to the cell membrane. Peripheral protein is a part of cytoskeleton which gives a structural integrity to the cell membrane. And there are another type of proteins known as transmembrane protein. Transmembrane, between the membranes. So they are the channels, carriers, pumps, and receptors. You have to know this, like which are all the transmembrane, which helps to transmit some substances from one cell to the other. They are channels, can call as channels, or carriers, which is carry something from one to another. Pumps, which will pump or which will give from low to high, which is pumping, or receptors from one cell to the or one part to other, the something is receiving, or receptors. So the transmembrane proteins are channel, carrier protein, pumps, and receptors. Coming to the cytoplasm is divided into ectoplasm and endoplasm. Keep it in your mind, the cytoplasm, the peripheral part of the cytoplasm that is situated just beneath the cell membrane is known as ectoplasm. Just beneath. And the inner part of the cytoplasm, that is interposed between the ectoplasm and the nucleus, is known as endoplasm, and which this cytoplasm contains many organelles. So the organelles are bound with a limiting membrane. Some organelles are not having a limiting membrane. So we can divide the organelles into two types, that is organelles with limiting membrane, organelles without limiting membrane. Okay, and the organelles with a limiting membrane are endoplasmic reticulum, Golgi apparatus, lysosome, peroxisome, centrosome and centrioles, secretory vesicles, mitochondria and nucleus. And only two structures are without limiting membrane. They are ribosomes and cytoskeleton. So we can remember the ribosomes and cytoskeleton as the organelles without limiting membrane, and all the other organelles are with limiting membrane. So ribosomes and cytoskeleton are the only structures without limiting membrane. So the functions of each organelles in a nutshell, let's see. The rough endoplasmic reticulum, which synthesize protein and help in the degradation of worn out organelles. The major function of the rough endoplasmic reticulum with means the endoplasmic reticulum having ribosomes in it. The ribosomes are attached into the endoplasmic reticulum, so which helps in this synthesis of protein. Smooth endoplasmic reticulum, which help in the synthesis of lipids and steroids. Also, there is a role in cellular metabolism and storage and metabolism of calcium. Also, catabolism and detoxification of toxic substances. And next, coming to the Golgi apparatus, which is a package unit. Remember, Golgi apparatus is a packaging unit. So, which will process, package, and deliver the proteins and lipids to the appropriate sites. The proteins and lipids synthesized in the rough endoplasmic reticulum and smooth endoplasmic reticulum will reach into the Golgi apparatus, and this one will package and deliver to the appropriate areas. Next, coming to the lysosome. Lysosome helps in the degradation of the macromolecules, or which degrade all these substances, and the degradation of the worn out organelles, removal of the secretory product, and perforin, granzyme, and melanin and serotonin will be secreted by the lysosomes. And next one is peroxisome. Peroxisome, which helps in the breakdown of excess fatty acids, detoxification of hydrogen peroxide and other metabolic products, oxygen utilization, peroxisome. So in the name itself, it is given oxygen. So it is related to oxygen. And that oxygen, or the oxygen utilization, will help in the breakdown of fatty acids or detoxification, acceleration of gluconeogenesis. Gluconeogenesis is the formation of glucose from other sources, and degradation of purine to uric acid, role in the formation of myelin, role in the formation of bile acids. And next one is centrosome. Centrosome in the center, so which movement of chromosome during cell division. Mitochondria, we know it is the powerhouse of cell, so which helps in the production of energy, synthesis of ATP, and initiation of apoptosis. Coming to the ribosome, which helps in the synthesis of protein, and cytoskeleton, which determine the shape of the cell, stability, cellular movement. The major one is the cellular movement. Cytoskeleton helps in the cellular movement. And nucleus, that is the major part which control all the activities of the cell. synthesize RNA, ribonucleic acid. Also, which the nucleus sends genetic instructions to cytoplasm for protein synthesis. And nucleus is the area which the storage of hereditary informations will happen from one generation to the next generation. The characters will go through the DNA. So the DNA is present in the nucleus. Let's see in detail about the cytoskeleton. Cytoskeleton provides shape and structure, help move organelles around the cell, and the major cytoskeleton materials are microtubule, actin filaments, and intermediate filaments. Microtubule, actin filament, and intermediate filament. And now we have seen about the cell. Let's see how the cells are communicating with other one, or how the cells are attached to the other cell. So the intercellular junctions are present between each cell. So there are the major junctions are tight junction, desmosome, and gap junctions. Tight junction, desmosome, and gap junction. Let's see what it is. Tight junctions are the one which give strength and stability to the tissue. In the name itself, it's given, it is tightly packed. It is having a very selective permeability, means it won't allow all the substances to pass through, which is having a fencing function, and which maintains the cell polarity, because it won't allow the mixing of the cytoplasm or mixing of the ions or mixing of the substances inside one cell to the other, and which provide the blood-brain barrier. Not only blood-brain barrier, blood-testis barrier also will be done by the tight junction, because it is just like a cement wall. It is clearly defined wall. It is wall. So the examples are epithelial lining of intestinal mucosa and renal tubule, endothelium in capillary wall and choroid plexus. That is the blood-brain barrier. Endothelium in capillary wall and choroid plexus. We have to remember the proteins involved in the tight junction. They are occludin, claudin, jam, singulin, symplecin, and izodo123. So the major proteins involved are occludin, and claudin. Even James and singulin are important, but major time we will remember about the occludin and claudin. Next one is gap junction. The name itself shows there is a gap between the two junctions, so which allow passage of small molecules, ions, and chemical messengers. There is no fencing. It's a gap. So through the gap, the molecules are passing very easily, and which helps the propagation of action potential. So the examples for the gap junctions are like in epithelial lining, heart, and intestine. Again, the protein involved is connexin. Remember, the gap junction protein involved are connexins. And next one is adherent junction. That is one cell to other cell by a cadherin molecule. There are filaments there are some filaments will be produced, and that will protein cadherin will produce filaments-like structures which will connect the cell to other cell, which is present in the epithelial lining, heart, and in the epidermis. Next, the fourth one is focal adhesion. Focal adhesion. Focal adhesion is the cell attached to the basal lamina and extracellular matrix. One cell attached to the basal lamina and extracellular matrix. That is the example of epithelium, epithelial lining of all the intestine and all. And this will provided by integrins. And fifth one is desmosome. Desmosome is again a cell-to-cell attachment. And it is also present in the epithelial lining and skin, which is the protein involved is known as cadherin. Cadherin. And hemidesmosome are also involved, or the protein involved is integrin. So remember, focal adhesions and hemidesmosomes are having integrin protein involved. So again, focal adhesion and hemidesmosome are not almost same, which will also provide attachment to the cell to the basal lamina and the extracellular matrix. And the transport mechanism. We have seen about the structure. There are the structure of the cell membrane, and all the organelles involved in it. Now, let's see how the digested food material will be transported from one place to other, or one cell to other. It's reaching all over our body. So how it is happening? So transport is one of the major life process in our body, because the nutrients has to be absorbed and it should redistribute all over our body, and the waste material should excreted from our body. Without this process, nothing can be happened in our body. So the absorption process is the first stage, which take the dissolved materials through the cell membrane to the interior. So substances are dissolved and that is taken inside the cell. That is the absorption procedure. And for that, the key membrane involved is cell membrane. For absorption, cell membrane is the major part for absorption. So the cell membrane which control what to enter and what to leave the cell, and that is why it is known as selectively permeable. So cell membrane is selectively permeable. It is not impermeable. It is selectively permeable. And cell membrane shows a fluid mosaic model. Cell membrane shows a fluid mosaic model which shows the proteins in the phospholipid bilayer, shows a mosaic-like appearance. So coming to the phospholipid in the cell membrane, the phospholipids having a head end and a tail end. The head end is known as hydrophilic head. Philic means loving. So water loving head and water hating tail. So the hydrophilic heads are attracted to the water and they are known as polar. The cell membrane, the phospholipid, the head end is polar and the tail end is hydrophobic in nature, which is not attracted to water, and they are non-polar. So the phospholipid bilayer proteins are attached, carbohydrates are there, and there are bilayer, means the tail ends are facing together and head ends are facing outwards. And the transport or the cell types of cellular transport, there are major three cellular transport mechanism. They are passive transport, active transport, and transmembrane transport. Passive, active, and transmembrane. Passive transport. Passive means cell doesn't use energy to perform the functions. They are the examples. Remember, the examples are diffusion, facilitated diffusion, and osmosis. There is no need of energy usage for these processes. And active transport, it is active, so the cell does use energy. Cell needs energy, means it is from a low area of a low concentration to a higher concentration. So the transmembrane transport, the cell uses energy to move substances. They are the protein pumps. And transmembrane transport, they are endocytosis and exocytosis, that is membranous transport. Again, the transmembrane transport also doesn't use energy. So the transport which uses energy is only active transport. Passive transport and transmembrane transport doesn't use energy. So if we have to think energy using and energy devoid of energy type of transports, active transport, primary active transport and the secondary active transport are the only transport mechanism which uses ATP or energy for their movement. So the passive transport, which move the substances or the molecules from an area of high concentration to an area of low concentration. High to low. So the movement of molecules are just flowing with the concentration gradient. There are three types of passive transport. They are diffusion, facilitated diffusion, and osmosis. Diffusion, which is known as the random movement of particles from an area of high concentration to an area of low concentration, high to low, until an equilibrium is reached. The rate of diffusion, it is depending on the concentration gradient. So if the high concentration area is mentioned as C1 and low concentration area is C2, so the difference C1 minus C2, C1 minus C2 is the steepness of the concentration gradient. Next, coming to the temperature. Temperature give molecules a kinetic energy, and energy is giving, so which moves around fast. So the diffusion is directly proportional to temperature. Again, diffusion is directly proportional to the concentration gradient. And surface area, if there is a large surface area, the substances will move faster. So area. Again, diffusion is directly proportional to surface area A. And the type of molecule or ion diffusing. Large molecules needs more energy. So large molecule or the shape of the molecule is also important. the circular substances or the circular molecules will diffuse easily comparing to the linear substances. If a substance is linear in shape, it will diffuse slowly, but if it is circular in shape, it will move faster. Non-polar molecule diffuse more easily than polar molecules. So, about some substances, the oxygen is non-polar, but oxygen is non-polar, so it diffuses very quickly. Carbon dioxide, in the other side, it is polar, but it is very small. Comparing to oxygen, the carbon dioxide is very, very small. The molecular weight is small, so it will diffuse very quickly. And water is polar, but it is very small. The weight is very small, so again it will diffuse very quickly. And diffusion through the protein channel, which depends on the selective permeability of the cell membrane. First one is simple diffusion. Now we have to see the diffusion through the protein channel. So which depends on the selective permeability. If the cell membrane allows, then only it will pass through. Next one is gating mechanism of the protein channels. There are voltage gated, ligand gated, and mechanically gated channels are present. Voltage gated means if there is a voltage difference, like an action potential, then only the gates will open. Voltage gated sodium channels. Voltage gated sodium channels are present at the neuromuscular junction. Ligand gated. A ligand is an enzyme like acetylcholine or GABA or whatever it is. A ligand has to attach to open the gate. And mechanically gated, a mechanical pressure or any change, like in the Pacinian corpuscle, a mechanical change will open the gate. So the voltage gated channels, the change or the voltage change, like sodium, the change in the sodium and potassium concentration will opens the gate. So the facilitated diffusion, it is the diffusion of specific particles through transport proteins found in the membrane. So the transport proteins are specific. They select only certain molecules to cross the membrane, and which helps to transport larger or charged molecule like glucose and amino acids without using energy. So, if there is a protein or a carrier protein is present, from this, it will move from higher concentration through a transport protein. That is known as facilitated diffusion. There will be separate carrier proteins will be available like GLUT, 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 or sodium dependent glucose transporter. Major part is GLUT. So what will happen is it will attach and it will help to move. Next one is osmosis. Osmosis is the diffusion of water from an area of high concentration of water molecule to an area of low concentration of water across a partially permeable membrane. High solvent concentration to low solvent concentration. That is the osmosis through a semi-permeable membrane. And the transport mechanism, if the transport mechanism, it is one substance is moving along with the other substance, that is known as co-transporter, or same means like the carriers that move more than one kind of molecule in or out of the cell. It is going together, that is known as co-transport. And if the molecules are moving at the same direction, it is known as symport. Again, that is also known as symport. Symport or co-transport. If the molecules are moving in the opposite direction, means one substance is entering and other substance is moving out, that is known as counter transport. So co-transport is moving together towards the same direction, and counter transport, it is moving in the opposite direction. So these are about the passive transport mechanism. Now let's see about the active transport. Again, energy is required and it is moving against a concentration gradient. Means a low concentration area towards a high concentration area. That is the uptake of iodine by thyroid glands. Because the thyroid gland cells are are rich in iodine, still we have to make the entry of iodine inside the cell, so which needs some energy. And if the molecules are moving only one kind of, if the transport mechanisms are moving only one kind of molecule, that is known as uniport. Uniport. And if it is in the both the direction, that is known as counter transport. The major example is sodium potassium ATPase, so sodium potassium pump. So there are two types of active transport. They are primary active transport and secondary active transport. Primary active transport, where energy is required to move a substance uphill. And the hydrolysis of ATP by the transporter provides the energy for primary active transport. And the major primary active transport mechanisms are, or pumps are, sodium, potassium, calcium, hydrogen ATPase, and hydrogen potassium ATPase. Major ones are sodium potassium ATPase, calcium ATPase, hydrogen ATPase, and hydrogen potassium ATPase. So the sodium potassium pump is the major some primary active transport, which is present in all of the cell, which helps in the maintenance of sodium and potassium concentration across the cell membrane, which helps to regulate the sodium potassium concentration across the cell membrane, which establish a negative electrical potential inside the cell, which establish a negative electrical potential inside the cell. So the sodium potassium pump exchanges 3 sodium ions for 2 potassium ion. Remember, 3 sodium ions are moving out and 2 potassium ions are coming in, which controls the cell volume and also the electrogenic activity of the cell. So a mnemonic to remember the sodium potassium pump, that is pump potassium in, 2 potassium in, 3 sodium out. So pump potassium in or pumpkin, pumpkin, pump K is for potassium in. Pump potassium in. Coming to the secondary active transport, the transport of substances are coupled to the primary active transport. So the transport use sodium. The primary active transport of sodium by the sodium potassium ATPase creates a sodium concentration gradient across the plasma membrane, which indirectly stores energy for the secondary active transport like sodium glucose symporter. There is a sodium will move from the high concentration to the low concentration, and there is a site for the glucose. So along with sodium, glucose are also enters into the cytoplasm. So in a nutshell, there are three modes of carrier-mediated membrane transport are there. They are facilitated diffusion, primary active transport, and secondary active transport. Exchangers are known as co-transporters or antipodes, co-transporters, and also there are sympodes. And the use of digoxin. Digoxin is the cardiac glycoside. What it will do is the digoxin and other glycosides improve the ionotropy by inhibiting the sodium potassium ATPase, and which increases the intracellular sodium level. So this increase in the intracellular sodium level weakens the calcium extrusion via the sodium calcium exchanger. So the calcium level will increase in the cardiac cell, and that will increases the myocardial ionotropy, and because of increased level of calcium, the cardiac output will be improved. That is the mechanism of action of digoxin. And the inflammatory bowel disorders like Crohn's disease and the ulcerative colitis are associated with increased intestinal tight junction leakiness and decreased expression of occlusion. So the tight junction permeability will be really decreased, and that will end in inflammatory bowel disorder. So the next, we are going to the that is endocytosis and exocytosis. Endocytosis is the condition where the cell is engulfing by using a bulge or the membrane bulge inward and forming a vesicle. So the phagocytosis is the type of endocytosis where entire cell is engulfed. Entire cell, a foreign body is engulfed. Pinocytosis where the fluid is engulfed. So commonly we use like a cell eating and cell drinking. Cell drinking or the pinocytosis is for the water molecule. And next one is the receptor mediated endocytosis, where some substances are binds to certain specific molecules in the membrane. One receptor, it is attached to the receptor. So exocytosis. So endocytosis is over, and exocytosis, which forces material out of cell in bulk. So there is a membrane surrounding the material which fuses the cell membrane, will release to the exterior of the cell like hormones and waste materials. So we I've seen about the cell, its cell membrane, and what are the transport mechanisms in the cell. Now, let's see how the messages are passing and how a message is formed in a cell and how it is passing. So, the message or the change is known as action potential. The brief sequence of change that occur in the membrane potential following excitation is called action potential. So let's see what will happen in the resting state and how the cell or how each cell is excited. Our body is made up of the basic unit cell. So each cell has to get excited to get the excitation of the group of cell, that is tissue, to the organs and the organ system. So let's see how it is happening. In normal case or in resting condition, the inside of the cell is negatively charged. Inside is negative and because large negatively charged molecules like proteins are present inside the cell, and sodium potassium pump which acts to causes the negativity. And also there is a voltage gated ion channels are present in the cell. So the cell is negatively charged inside. So resting potential, when neuron or cell is at rest, that sodium concentration is more in outside, potassium concentration is more inside, and negatively charged proteins are found inside the cell. And the outside is positive and inside is negative is known as polarized condition. Remember, if a cell is polarized, means the inside of the cell is negative and outside of the cell is positive. So for neurons, there is a voltage difference of minus 90 millivolt. The resting membrane potential is contributed by potassium diffusion potential, sodium diffusion potential, and sodium potassium pump. So the diffusion potential is potassium outside it is 4 milliequivalent and potassium inside is 140 milliequivalent. And coming to the sodium, outside it is 142 milliequivalent and inside it is 14 milliequivalent. Okay, so just make it together. Outside it is around 146. It is an approximate value, but still sodium is 142 and potassium is 4 milliequivalent. And inside, again, sodium is 14 milliequivalent, only 14, and potassium is 140 milliequivalent, which provides a negative current of around minus 94 milliequivalent, or sorry, millivolt. And along with the negative charged protein molecule, it produces a negative potential inside the cell. So the resting membrane potentials of different excitable tissues like myelinated nerve, it is minus 90 millivolt. Average myelinated nerve, it is minus 70 millivolt. For a skeletal muscle, it is again minus 90 millivolt. And for ventricular muscle fiber, it is minus 90 millivolt. And for atrial muscle fiber, it is minus 85 millivolt. And Purkinje fibers, it is minus 95 millivolt, and pacemaker tissues, it is minus 65 millivolt. And the smooth muscle fiber, it is varies from minus 50 to 60 millivolt. Minus 50 to 60 millivolt. So the lowest membrane potential, resting membrane potentials are in the the smooth muscle. Pacemaker tissues are the AV node and all, and the smooth muscle. So the resting membrane potential, the neuron is said to be polarized. That is negatively charged on the inside and positively charged on the outside. And this polarized state will maintain until a neuron is stimulated. So the ability, not only neuron, the ability of a cell to respond a stimulus and convert it into a nerve impulse is known as excitability. So from a polarized state, if the neuron is respond to a stimulus and it convert to an impulse is known as excitability. And we are talking about neurons here, because neurons are the one which send impulses to all over the body. So a nerve impulse which starts when a pressure or other sensory inputs like pressure, thermal, chemical, or any sensory inputs which disturbs the neuron's plasma membrane, which causes sodium gates to opening of the sodium gate. So when the sodium gates are open, which allow positively charged sodium ions to flow inside the cell membrane. And the inside of the membrane is temporarily more positive than outside. That is known as depolarized state. Depolarized state. And when the impulse passes, the potassium gates open, which allow the positively charged potassium ions to move out. That is known as repolarization. That is regaining of the normal polarized state. And the rapid depolarization and repolarization process generates a voltage pulse peak that is known as action potential or nerve impulse. So, if we see this action potential, when at resting state, both the sodium and potassium channels are closed, and at a threshold level, a stimulus opens some of the sodium channel, and if the sodium influx achieve a threshold potential, there are more sodium channels will open. First, there is a slow opening of the sodium channel. Once it is a minimum requirement, there are so many sodium channels will open. And that causes the depolarization, activation of gates of the sodium channel. And the potassium gates are closed during this period. Only sodium channels are opening. So the sodium ions are moving inside the cell. And the repolarization phase, that is at the peak, there is opening of the sudden closure of the sodium channel and slow opening of the potassium channel will happen. So the opening of potassium channel leave the cell and lose the positive charge, causes the inside of the cell to become more negative than outside. So again, it is the remember the potassium channels won't close fast. It is having a slow closure. And the sodium channels are closed, but the potassium remain open. So the resting state, it has to reach the resting state, but before that it will go below the resting potential, that is known as after hyperpolarization or undershoot. So these are all the phases of an action potential, that is resting membrane potential, stimulus artifact, latent period, firing level, depolarization, overshoot, repolarization, after depolarization, and after hyperpolarization. So the stimulus artifact, which is due to the leakage of current from stimulating electrode to the recording electrode. This is what we are going to record. And latent period, that is the interval between the application of stimulus and onset of action potential. Remember, it is around 0.5 to 1 millisecond. 0.5 to 1 millisecond latent period. Firing level, that is the depolarization proceeds up to a level of 15 millivolt, known as firing level. From there means there are opening of the more number of sodium channels, or so more number of sodium channels. So there is a sharp peak, that is known as firing level. Depolarization, that is around 7 to 10 millivolt. Voltage gated sodium channel open in a faster rate, that is known as sodium channel activation, that is up to a voltage of 7 to 10 millivolt. So the firing level is 15 millivolt. There is more sodium influx, inward movement of the sodium will happen. Overshoot is from the firing level. The curve reaches a zero potential rapidly, and it will reach to plus 35 millivolt. It is starting from the minus or negative, and it is reaching the positive of plus 35 millivolt. That is because of the influx of large amount of sodium. The spike potential, that is the phase of rapid rise of potential in the depolarization and rapid fall in repolarization, that is around 1 millisecond. That is because of the closure of sodium channel, that is sudden closure of sodium channel and opening of the potassium channel. Coming to the repolarization, that is also known as negative after potential, that is around 4 milliseconds. And that is due to the opening of potassium channel. And after depolarization, that is slow fall in the membrane potential. At the end of repolarization is after depolarization. That is the last phase of potassium efflux. That is the spike potential. Potassium conduction is slowed down, and a few milliseconds are delayed in the restoring the membrane potential because it is slowly closed. The potassium channels will close very slowly. And after hyperpolarization, that is after reaching the resting level, there is a further fall and become more negative. That is also known as positive after potential. It reaches around 35 to 40 milliseconds. Finally, the resting membrane potential is restored by the action of sodium-potassium pump. So coming to the properties of action potential, the action potentials having a threshold stimulus, which shows an all or none response, refractory period, conductivity, and accommodation. Let's see. A minimum current which produces an action potential is known as threshold stimulus. There is a strength duration curve we can draw. In that, we have to remember 3 points. That is, Rio base is the minimum strength of stimulus which can excite the tissue, whatever may be the duration. Rio base is the minimum current we are giving and which is exciting a tissue. And utilization time is the minimum time required for the minimum current or the rheobasic current to excite the tissue, how long it is taking. And chronaxy is the minimum time at which the double the rheobasic strength. We are giving the double the rheobasic strength voltage and how long it will take to excite the tissue. These 3 points we have to remember. In that, chronaxy is an index of excitability of the tissue. So we can use chronaxy to compare the excitability of various tissue. So longer the chronaxy and lesser the excitability. All or none response shows that an action potential fails to occur if the stimulus is subthreshold in magnitude, or it responds to the maximum. If we are giving a threshold stimulus, it will give its maximum. Refractory period, that is the duration by which the cell remain refractory or non-responsive to the second stimulus, is known as refractory period. There is an absolute refractory period. During this period, no stimulus will produce a response, irrespective of the strength. And relative refractory period, the stimulus with stronger than normal can cause excitation. So, absolute refractory period falls in the depolarization phase and relative refractory period may falls in the repolarization. Conductivity. The action potential produced at one location acts as a stimulus for the production of action potential in the next cell. Means it is moving. It is like a wave of depolarization. So the factors affecting in conductivity are temperature, pressure, blood supply, chemical, pH, ion, size of the nerve, and myelination. So how myelination is helping in the propagation of action potential? So the voltage gated channels in axon open, serving to propagate the action potential. In myelinated neuron, there is a condition known as saltatory conduction that speeds the conduction velocity. So, which conduct the impulses faster, and conduction velocity is proportional to the diameter of the axon. Diameter, that is, larger neuron will move faster or they will conduct the impulses faster. And the myelination, which allow the small diameter axons to conduct signals fast, that is known as the saltatory, means jumping of the impulses from one node to the next. So, one node of RAN wire depolarizes the next, so the action potential will skip. So, that is known as saltatory conduction. And accommodation, that means if an excitable cell is submitted to the passage of a constant strength of current, the site of membrane under the stimulation fails to produce, that is because it is adapted. We are giving same strength of current because of the slower opening and delayed closure of the voltage gated potassium channel. So accommodation is happening because of the characteristic feature of potassium channel. So by this, we can finish today's topic, and let's see in another class. Thank you.
Thank you.